The COVID-19 pandemic has highlighted the importance of vaccines in protecting against infections or in reducing the severity of symptoms among vaccinated individuals. Vaccines typically contain a harmless form of a virus or bacteria known as the antigen. Upon vaccination, activated T cells provide help to B cells, which go on to differentiate into antibody-secreting plasma cells. These antibodies are key to conferring protection against an infection due to their ability to neutralize virus. Vaccines are also important in generating memory cells, which can generate a faster and more potent response during an actual infection. Vaccine-induced antibodies can arise from two main pathways. Antigen-activated B cells can differentiate into short-lived plasma cells early in the immune response via the extrafollicular pathway. Alternatively, B cells can enter the B cell follicle where they undergo class switch recombination before entering the germinal center response. The germinal center is a specialized microenvironment which is composed of two compartments. The dark zone, where B cells undergo clonal expansion and somatic hypermutation of their immunoglobulin variable genes, and the light zone, where B cells receive antigen from the FDC networks and undergo a process of selection. B cells that receive positive selection signals from T follicular helper cells will either re-enter the dark zone for further proliferation and somatic hypermutation, or exit the GC and differentiate into long-lived antibody-secreting plasma cells or memory cells. Unfortunately, during aging, vaccine efficacy is reduced, which means that the vulnerable members of our population are also the least protected. This age-related decline is characterized by a quantitative decline in vaccine-induced antibody titers, qualitative decline in terms of reduced antibody function and reduced diversity of antibody genes. In order to find ways to improve vaccine responses in older people, the first step will be to understand key contributing factors underpinning this phenomenon. Multiple mechanisms underlying the age-related decline in humoral response have been characterized, including defects in T cell priming by aged dendritic cells and dysregulation in stroma function. However, whether B cells intrinsically are defective in their function with age remain unclear. In this paper, we aimed to investigate if there are cell-intrinsic defects in B cells' response to stimulation or vaccination with age, using a combination of human in vitro studies and mouse in vivo studies. To understand if B cells from older people are as functional as those from younger people, an in vitro assay was set up when naive and memory B cells sorted from the peripheral blood of younger and older people were cultured with T cell signals, namely CD40 ligand and IL-21. After several days of culture, we compared the functional capacity of B cells from younger and older people in terms of their ability to proliferate, differentiate into plasma cells, secrete antibodies, and upregulate co-stimulatory molecules. After 6 days of stimulation with CD40 ligand and IL-21, we found that naive and memory B cells from older people do not have defects in proliferating, as shown by similar percentages of cells in each division and the similar average number of divisions undergone per proliferating cell, also known as the proliferation index. We also observed that naive and memory B cells from older people did not have intrinsic defects in differentiating into plasma cells 6 days post-stimulation with CD40 ligand and IL-21. Next, we compared the antibody-secreting capacity of stimulated naive and memory B cells of younger and older donors. To determine the antibody-secreting capacity on a per-cell basis, we determined the antibody concentration using ELISAs and normalized them with the numbers of antibody-secreting cells in each well. We observed that stimulated naive and memory B cells from older donors have similar antibody-secreting capacity as those from younger donors, showing that the plasma cells derived from B cells from older people are equally functional as those derived from B cells from younger people. To further probe the ability of B cells from older people in responding to external stimuli, we stimulated naive and memory B cells from younger and older donors using different combinations of T-dependent and T-independent signals, namely CD40 ligand IL-21 with IL-2, CpG, which is a toll-like receptor 9 agonist, and CD40 ligand with CpG. Across all conditions, no age-related defects was observed in plasma cell differentiation. IL-21 was also shown to be key in promoting plasma cell differentiation and stimulating the B cells with CD40 ligand alone or CD40 ligand IL-2 yielded minimal differentiation, consistent with previous reports. No significant defects in antibody-secreting capacity were also observed in naive and memory B cells from older human donors across all stimulation conditions. Together, this demonstrates that B cells from older people do not have intrinsic age-related defects in differentiating into functional antibody-secreting plasma cells as compared to B cells from younger people. Upon activation, B cells increase expression of co-stimulatory molecules, which are important for the activation of T cells, and reciprocally for B cells to receive survival and differentiation signals during T-dependent responses. After 48 hours of stimulation with CD40 ligand and IL-21, naive and memory B cells from older people were generally able to upregulate CD80, CD86, HRA-DR, and IL-21 receptor to similar extents compared to their younger counterparts. We also observed that stimulated naive B cells from older people had a higher expression of HRA-DR, which is consistent with another study that reported higher expression of HRA-DR in the PBMCs of older people, although the impact of this on B cells' function in aging remain unknown. To conclude our human in vitro studies, we found that naive and memory B cells from older people were not defective in proliferating and differentiating into functional plasma cells. In addition, B cells from older people are able to upregulate co-stimulatory molecules to similar extent as those from younger people. This suggests that there are generally no intrinsic defects in B cells from older people in responding to stimulation. We then went on to investigate if there are cell-intrinsic defects in B cells' function in vivo using the switched HEL adoptive transfer system. Hennick lysozyme or HEL-specific B cells from either a young or aged switched HEL mouse were transferred into young congenically distinct wild-type recipient mice. These switched HEL B cells contain the high-affinity B cell receptor against Hennick lysozyme. The recipient mice were then immunized with HEL conjugated to straight red blood cells to elicit a T-dependent response, and the response mounted by the transferred B cells were analyzed at days 3, 6, and 10 post-transfer and immunization. Similar to what we observed in the human in vitro studies, B cells from the aged donor mouse had no proliferative defects at day 3 post-immunization compared to those from the young donor mouse. During immunization, activated B cells can undergo 3 main cell fates. Early post-immunization, some activated B cells can migrate to the extrafollicular regions and differentiate into short-lived plasma cells. Alternatively, activated B cells can enter the GC reaction and eventually differentiate into long-lived plasma cells or memory B cells. To analyze the impact of B cells' age on these pathways, we analyzed the cell fates of transferred B cells in recipient mice at days 6 and 10 post-immunization, where the extrafollicular response and the GC response were approximately at its peak, respectively. For both time points, we did not observe any significant differences in the percentage and numbers of HEL-binding donor B cells we could recover in the spleens of the recipient mice. This suggests that B cells from aged mice are unlikely to have defects in their survival in the recipient mice and in homing to the spleen. At day 6 post-immunization, B cells from the aged donor mouse preferentially entered the extrafollicular response, as shown by the higher percentage and numbers of plasma cells compared to donor cells from the young mouse. Significantly lower percentages and numbers of GC B cells were observed among transferred B cells from aged compared to young donor mouse. This resulted in a significantly reduced GC B cell to plasma cell ratio among donor B cells from the aged mice compared to those from the young adult mice. Together, This suggests that transferred cells from aged mice preferentially enter the extrafollicular response at the expense of initiating the GC reaction. At this time point, we observed no significant differences in memory B cells formation by transferred B cells from young or aged donor mice. We then performed confocal imaging of the fixed spleen sections at this time point to compare the localization of the donor cells from young or aged mice. Corresponding to what we observed in our flow data, HEL-positive CD45.2 positive donor B cells from the aged mouse were more clearly observed in the extrafollicular regions of the spleen compared to those from the young mouse. Conversely, HEL-binding donor B cells from the aged mouse in the GC appeared to be more sparse compared to those from the young mouse. Together with our flow data, this suggests that B cells from the aged mice preferentially enter the extrafollicular response and have a delayed early GC response compared to B cells from the young mouse. Next, we looked at B cells' response day 10 post-immunization when the GC response is more mature. Here, we observed no defects in the ability of B cells from aged mice in mounting a GC response. This suggests that while age-related intrinsic B cell changes delay the GC response, they are unlikely to be responsible for the impact antibody-secreting response or smaller peak GC response in aging. We also observed no age-related cell-intrinsic differences in class switch recombination by B cells from the young and aged mice. While this is consistent with some studies, other studies have reported reduced class switch recombination by B cells from aged mice and humans due to lower expression of the AID enzyme. This may suggest some differences in B cells' response in vitro and in vivo, and/or that there are some factors in the young microenvironment in vivo, such as lower levels of inflammatory cytokines, that can compensate for the reduced expression of AID in aged B cells linked with impaired class switch recombination. As our data have shown that aged B cells do not have intrinsic defects in proliferation or differentiation into GC B cells in a young environment, we sought to test the hypothesis that B cell extrinsic factors contribute to the age-related defects in GC response by conducting another transfer experiment. Here, HEL-specific B cells from young donor switch-HEL mice were transferred into young or aged wild-type recipient mice. 6 days post-transfer and immunization, we observed a significant reduction in the recovery of young donor B cells in aged recipient mice. We also observed a reduced number of HEL-binding GC B cells by the young donor cells when they were transferred into aged recipient mice compared to when they were transferred into the young mice. In conclusion, in our paper, we show that B cells from older people are not defective in activation, proliferation, and plasma cell differentiation. In addition, B cells from aged mice showed a delay in GC B cell formation early in the immune response, but had no defects in mounting a peak GC response. We also observed a reduced recovery of B cells from young donor mice when they were transferred into aged recipient mice, resulting in a smaller GC. This implicates B cell extrinsic rather than B cell intrinsic factors as key contributors to the age-related decline in humoral response during vaccination.